Good afternoon. Well, I didn't realize I was going to be introducing the front page news. Montana Standard, which is Alysia said she hasn't seen the article, although she knew it came out today.
I grabbed it this morning and then was like, oh, gotta go.
To me, it was a really nice article. Maybe that's part of the reason some of you were here and found out about it. So it's a nice summary of what's going on. But it's I'm standing in for Bev to introduce Alysia today. She's an assistant professor of environmental chemistry. She grew up in Michigan. Where in Michigan?
Just south of Ann Arbor, in a little town called Saline.
Okay, south of Ann Arbor, and then made her way to Arizona State University, where she got her BS in geological sciences with minors in biology and German. She earned her PhD in chemical oceanography at Massachusetts Institute of Technology, otherwise known as MIT, and did some postdoctoral work at Arizona State University in the Swiss Federal Institute of Technology. What she's going to talk about in the paper today, she was just before Christmas, she went on an early career scientist training cruise, which she was selected from a group of the paper articles, 14 or something.
It was 50-something, and there were 11 of us.
11, that's right. 11 people ended up doing this, and the results of that work, I think, is what she's going to talk about today. So. The title, which is Rocks, Hot Water, Microbes, and More, Diving to the Seafloor Hot Springs. So, thank you.
Okay. Thank you very much. I'm very happy to be talking to you all about this today. This was something I had always wanted to do my entire life, sort of thing, that I got to go to the ocean floor. So I'm going to be telling you about my experience and then showing you a little bit of data, and hopefully you will all be as excited as I am. So I'm going to pass some things out. There are a few flyers here that talk about the vehicles that we used. Alvin is what we went down in to the seafloor. Sentry is this great vehicle, robotic, operated from the ship that goes down and makes really good maps of where you're about to take Alvin to. So that's what Sentry is. And then a little info about mid-ocean ridges. So I'll start these up here with Phil and pass those around. Remember, down there at the seafloor, 2,522 meters deep was how deep our dive went, which is about 8,200 feet. So a mile and a half down below the seafloor. There's a lot of pressure. So outside the sub is well, I didn't bring any of the colored ones, but you draw on your styrofoam cup, you sign it, and then you send it outside the sub, and it shrinks like this when you come back up. That's how much pressure there is down there. But me, my other science observer, and the pilot are preserved by a 3-inch thick titanium metal sphere, which is rated to go down to these pressures. So I'm also going to pass these around. I'll start these over here. Okay. And the other thing that I'm going to pass around is a tubeworm casing from the bottom of the ocean. The biologists, they took out the worm part and dissected it and did whatever with it and preserved it and are studying it, and they were like, oh, well, we don't actually need the tubeworm casings, so they handed them out, and so I can use them for outreach events like this. So the worm was in here. You'll see nice images of the worms, and this is what it's like. So we'll start that over here as well.
Dried, or has it been preserved?
It was preserved a little bit, yeah. Yep, and dried out as well. And then I have a rock up here, which you'll see, and I'm not going to pass this around, but people can come and look at it later. And it's this right here. Oh. So this piece right here is right here, and we will cut it up a little bit and look at some of the mineralogy. Chris is going to help us with that here. Kyle Nacy, a master's student, is going to work on this project. And then we'll probably bring it over to the Mineral Museum, But first I want to show it to people. So that rock is from right here. There's some diffuse hydrothermal flow coming up, precipitating out all of these minerals here that you'll see. They're sulfide minerals right there. Okay, so this was an early career scientist cruise, as Dr. Gammon told you, and it is sponsored by the UNOLS, the United National Oceanographic Lab System, on ships, and it is a Navy ship. So the ship is owned by the Navy, but operated by the Woods Hole Oceanographic Institution. So we go out on these boats, and the funding is mostly from ONR as well as NSF. So scientists apply for funding from NSF to use these sorts of ships. All right, so let's get going. There she is. This is the RV Atlantis, in port in Manzanillo, Mexico. So she is one of a couple of sister ships. She's 274 feet long, 52 feet across, and she's the only ship in the fleet that is able to have Alvin, the human-occupied vehicle, the deep submergence vehicle that we go down in. What makes it special with Alvin is there's a nice hangar to put Alvin in. There's some tracks, as we'll see, for Alvin to get out to the back, and then an A-frame and a really awesome rope, which we'll see a picture of as well, to take Alvin out behind the ship. Okay. When we got back to port in San Diego, we gave tours to the public of Alvin. So you're kind of getting a little tour, but I don't have the Atlantis here for you today, and I don't have Alvin for you today. So what I do have are images of this. So here I am giving a tour in San Diego of the Atlantis. Okay, this we're going to click on. These are two gentlemen that work with the Alvin crew. So it takes an entire crew of people to do this on the ship. There's the crew, and there's the Alvin crew who work with Alvin here, and then the Sentry crew who work with Sentry, part of that team, and then the science party. So this is Alvin here, and the titanium sphere is in the front part, and then all the rest of this white stuff is for moving Alvin around, as well as a syntactic foam for ballast for coming back up from the seafloor. Okay. So there she is. And you get in and out right there off of the red part, as we'll see. Okay. This is Alvin with her skins off there. So here you can see inside, every single one of those tubes has hydraulic fluid in it. Every single part of this is rated to go down to 4,500 meters. The sphere is actually rated to 7,500 meters, or sorry, 6,500 meters. And this is the second version of Alvin. She started operation in 1964, and was re-outfitted in 2013, and is going to be outfitted again in 2021. At that next outfitting, all of this stuff is going to be rated to 6,000 meters. And once it's rated to 6,500 meters, that's 98% of the seafloor. 4,500 meters right now is like 66-ish% of the seafloor. So Alvin can explore 66% of the seafloor right now. I like all the hydraulic fluid. It's really cool. So. Every single part on this ship is tested and rated to go down. So you can't, when I got to go down, I wasn't allowed to bring my cell phone or anything, and everything has to be tested for toxicity and go through the most rigorous standards, and the Navy has to give the final approval, for example. Okay, there's Alvin in the hangar, and this is at night. Alvin goes out during the day. So deployed in the morning, on the seafloor all day, and back out in the evening. And then at night, everything gets double checked, and then everything gets triple checked the next morning. And at night, this is the science basket here. All of the gear that we're bringing down to do science on the seafloor. This cruise was a two-part cruise. One of it was a makeup cruise for a part that got canceled because they were out in the middle of the Pacific Ocean and somebody had appendicitis. And they had to book it to Mexico to, not have somebody have their appendix rupture in the middle of the Pacific Ocean. So the first 4 dives on our cruise were at Seamounts, and the second 4 dives were for our early career scientist training at the East Pacific Rise at 9 North. So here we all kind of are standing around at night getting our basket arranged for Alvin. And on this basket is something called a slurp. It's basically a vacuum that sucks up crabs or rocks or things from the seafloor. There are bio boxes for putting in crabs and tubeworms and bringing them up for the biologists to study. There are major samplers for sucking out hydrothermal fluid at the seafloor. And what else? Those are some of the big things that are on there. Some Niskin bottles. So those can just get activated, and they are a discrete sample from a particular depth as well. And all of that goes on the basket of Alvin and goes in. These are these tracks that Alvin is wheeled out on to the back deck. Okay, so what happens to go out? Well, you climb up these steps, and here you take your shoes off and put them in this bag. They don't go down with you. You make sure you have enough warm clothes. They're like, oh, it's going to be cold, like 50 something. I'm like, ha. We live in Montana. That's not cold. Because remember, we're in the eastern tropical, Pacific, and it was really warm out. But anyway, I did put on a sweatshirt down in the sub. So you pack your little go bag the night before, and they look in and make sure it's all okay. And you bring a pair of socks, and you bring a sweatshirt, and then you're pretty good to come back up. Clipboard, pencil. All right, there we are. All right, so now Alvin is going to be deployed. So wheeled out to the back deck, hooked up to the A-frame, and that is that big rope I was telling you about. So Alvin weighs 40,000 pounds, and that rope is rated to 280,000 pounds, so 7 times Alvin. Yeah, so it's okay. It's gonna be all right. So Alvin is hooked at this point directly to the A-frame and to the rope. So the A-frame is weighted with Alvin, and then it's gonna be deployed out the back, and then the A-frame unhooked, and then it's on the rope, and then it ends up being unhooked from the rope, and then the swimmers are going to go around and do double checks on Alvin, communicate with the pilot. The swimmers go off in the Zodiac, make sure that they're far away from the sub, and then the pilot does more checks, and then you go down. So let's deploy Alvin. Okay, so I'm standing up on the deck taking pictures of this deployment. So here we go. Alvin's lifting up. Woo! Down in the water. Look at that. So detached from the A-frame and on the rope. A little bit swinging around there into the water like that, and then detached from the rope. So there's the big rope that can hold 280,000 pounds. It's like this big. It's really quite a rope. Okay, so now Alvin is out there. You can see the Zodiac over here, and then the swimmers are going to go, once the Atlantis is far enough away, the swimmers are going to do all their checks, and the swimmers will get in the Zodiac, and then Alvin will go down. Okay. All right. This is a key point. I love this when they do this. So the swimmers, they are double checking to make sure that Alvin has come off of the A-frame and waited on the rope. And so they both do the thumbs up, and it's a really epic photo. And Rika Anderson from Carleton College had a very nice camera and was taking very excellent photos of this, and that is from her. So Rika took this picture of Alvin being, yes, we're ready to go. We can weight the rope and put Alvin out the back. Okay, in the sub. So now you're in the sub. It's a little bit fuzzy, not the best picture, but I just wanted a picture from in the sub. And you have a couple of a meter and a half or so of space. So I would be right here, the pilot would be right here, and then Elizabeth, my co-science observer, is just on the other side. And so we have a camera in there that we can take pictures out of the windows, and she took a picture of me like this. We have dark red light, all sorts of equipment. We have safety equipment over here in case something ever went wrong as well. All right, so then we're down all day, and then we'll talk about that in a minute. But then at the end of the day, the ship knows where Alvin's going to come up, and we go to kind of near where Alvin comes up, and you go out to the bow, and you stare off, and you look, and then, oh, there it is, it pops up. And then this is a dude, a swimmer, talking on a phone. It's a literal old handset phone, and he's talking to the pilot in the sub. And it's really funny, because there's like these guys, in their their swim shorts and no shirt, just chatting, all the way down, and Alvin goes all the way down the side of the ship, all the way aft, until we bring Alvin back on board. Okay. This is another photograph from Rika Anderson, where Alvin has gone all the way to the back, and now we're hooking Alvin back up to come back up on the ship. So they can actually deploy and recover Alvin in a certain degree of waves, and it actually works out all right. So this isn't actually very wavy at all, but they're recovering Alvin. Okay, so what did we actually do on this? So we went from Manzanillo, Mexico. We transited down to 820 Seamounts, and these Seamounts are really actually interesting because they had thought that the rock composition was going to be the same across here, and then there was some evidence that it actually wasn't, and it's actually not. The rock composition across here is actually very, very different, so that's what they were working on. And then, so there were 4 days of dives here, and then we did our East-Pacific rise dives at hydrothermal vents, there. Okay, so these files are really big, so I had to put them on the desktop, and you're going to see the best of some of the stuff on the seafloor here. And let's go to this. Okay. So this is taken with a GoPro camera from that is attached to Alvin. This is not from my dive. This is from a dive after, because this particular camera wasn't working on our dive. But these are the basalts. So remember that the East Pacific Rise is an ocean spreading center. It's where ocean crust is being formed. It's basaltic rock. Another type of basaltic rock is Hawaii and Iceland. Those are also basaltic rocks. And so this fresh, shiny surfaces, that is relatively newish rock. The last eruption at the East Pacific Rise was in 2005, 2006, and the one before that was about 15 years before that. So there are eruptions here about every 10 to 20-ish years. And one of the things that we were looking for was if there was a new eruption. Because when there's a new eruption, it restarts the ecological community that's living down there. It changes the plumbing of the hydrothermal fluid systems a little bit as well. But there wasn't a new eruption when we came out there this time. So you're seeing some very nice seafloor basalt. You're seeing also these cavities. And these cavities are where there was actually vapor underneath when this rock cooled, and then later it kind of collapsed. And actually, Alvin comes up with drop weights, and when you drop the weights a little bit off axis, it kind of crinkles through the basalt, and you can hear like crinkling glass, kind of. So that's basically, the glassy stuff is basaltic glass composition. Aha, okay, so now we're coming up on an event here with this. That little flash that you saw is a camera underneath Alvin that is taking a picture of below once every 10 seconds. I'll take you through some of ours from our dive as well. So what you're seeing here are sulfur as our hydrothermal fluids are coming up and exiting. You'll see hydrothermal fluids that are kind of shimmering. That is a diffuse flow. So the seawater is 2 degrees Celsius, just above freezing, and the hydrothermal fluid is warmer than that to hundreds of degrees, and then we'll soon see a picture of a black smoker, and the black smoker is like 350 Celsius. And so we have hot, metal-rich, anoxic hydrothermal fluid coming out, meeting cold, oxic seafloor, seawater. And there's a crab in the upper left, and then that shimmering is the diffuse flow coming up. So on this trip, the 11 scientists, we were geophysicists and microbiologists and geochemists and macrobiologists, and so we collected coordinated samples for all of these things down there. There's a tubeworm over here, yes. So this is a tubeworm, just like the one that you're passing around. It could have been that one. No, I don't know which one it is. Yeah. So those big tubeworms, those are called giant tubeworms. Those are Riftia is the genus, and then the species starts with a P. And then there are some little ones that are Alvinella. Where are they? Don't see them yet. They look like little flowers. This looked like a hole for an Alvinella tubeworm as well. So again, more shimmering. You'll see the tail of a polychaete worm. It kind of came, oh, here we go. That's an Alvinella tubeworm. Here, a little one. This is a polychaete worm that kind of danced in front of the camera, so it's kind of putting on a show. How big is that worm? Oh, so I wasn't on this one, but I think that worm was like this long and like this wide. Because so here's Alvin's arm, and then we collected a bunch of our samples through this tube as well. Maybe it's actually a little bit longer than that. And then that rope was from one of our microbe traps from one of the scientists as well. More nice diffuse flow coming out from the seafloor. So this is placed right now to some kind of epic music, but you have to listen to me instead, There we go. That's the black smoke, okay? So the minerals are forming as the fluid is coming out and touching the seafloor. So the temperature gradient is from 350 to like 2 Celsius in a very small space. That's what we're seeing there. There's a fish. I probably should have looked up the name of the fish, but that's a fish. It has eyes, but remember, there's no light down there, right? So as you're in the sub and you go down, it gets dark, and then it's pitch black as you're going down. It takes about an hour to go down to how deep we were. And then we had about 6.5 hours of bottom time, and then an hour to come back up. And then plus the deploy and recover times, which is like half an hour depending on, what's going on. So yeah, there's the black smoker part. Aha, so here we go. Here's some tube worms. And these are some mussels, and then there are crabs, and then there are squat lobsters as well. Where's a squat lobster? So the squat lobsters are shaped a little bit differently than the crabs. There's a good image of the tube worms. Notice how that one is retracted up here. So crabs eat tube worms. So one of the scientists studies crabs, and we had to put down a crab trap with some tube worm parts to trap the crabs. And if you're down there and you see a crab going towards a tube worm, it goes into its tube to get away from the crab.
Those are little shells on the outside.
Yes, and these are little shells also on the outside. Yes. So these are all fueled by chemicals from the seafloor. So hydrogen sulfide oxidation is pretty much driving a lot of this ecosystem. These tube worms have a symbiotic relationship with bacteria that are actually oxidizing sulfide in the tube as well. There's some more fish looking pretty good as well. Oh, so the other great and new thing about this is these GoPro cameras. So Dr. Dan Furnari, he's an emeritus scientist at the Woods Hole Oceanographic Institution. Part of where I did my PhD was joint with MIT and Woods Hole. He is getting these really good cameras put together and rated to go down. And so this is some of the best footage that you'll ever see of the seafloor so far, because no one's ever actually taken 4K video before of the seafloor. So all of the video before is a little bit fuzzy. So this is amongst the best. It's really good. This was put together from the dive by an artist that came along with us.
Is the camera on this side of the pressure or on the other side?
It's in the pressure. Yeah. Yep. So it has to get rated, and it's in some sort of a case, and Dan would keep it cool, and then he would run it out right before the deployment and then secure it on, and they would double check it, and then it went down because they were having problems with fogging and things with changing temperature as well for these cameras. So this is actually a GoPro camera for this. Yeah.
How much does it shrink?
Yeah, it's in a nice case. But who knows, maybe that's why it wasn't working on our dive. All right, and so then that was some of the really cool footage from some of the vents. And we'll see more from my dive as well. And then here's some of the nice shiny basalts. Come on. Ooh, I had no idea what that was. It's probably having a little bit of trouble playing all this video. This is not the 4K, this is the 1080p. Okay, and we'll look at all of the thank yous in a bit too. All right, so that's a little bit about what it's like actually on the seafloor. All right, so as I was saying, it takes a whole crew to go out there and do all this work. This is the science party and the Alvin crew and the Sentry crew. And so the ship's crew aren't even on here. There were 56 people on board, 53, something like that. So nothing gets done without everybody working together. So there's Alvin, and then this is Sentry. Sentry goes down and makes really awesome detailed maps. And then if something is interesting, you can send an Alvin Dive down there. They can actually turn over a subset of the data. Sentry goes down at night. So during the day, the only thing that's happening is Alvin is down on the floor, or on the seafloor, right? That's it. No CTDs, no collecting other samples, no Sentry, no nothing, just Alvin. And then at night, Sentry goes down and makes maps, and then you can do CTDs, which are conductivity, temperature, depth, collecting water from the water column as well. So there we all are, out there doing this. There's Sentry getting recovered and deployed off of the side. Gosh, I think Sentry weighs 2,000 pounds-ish, I think. And Sentry goes and hovers, I think, 40 or 50 meters above the seafloor and makes good maps, just like this. So when you're up on the top and you're doing multi-beam bathymetry, using waves to get your seafloor, it looks like this kind of fuzzy stuff. So you have an idea of what's going on with the depth. But when you send Sentry down to make a map, you get these really detailed swaths. And if we had put Sentry's path on here, Sentry is programmed remotely. They keep an eye on it from the ship, and it goes in patterns like this. The line that you're also seeing on here is one of the Alvin Dives 5003. We have 2 push cores from this dive here in the freezer at Tech. So I don't actually remember, I didn't look where they're collected, but whenever they saw a sample or sediment, they actually collected a push core. So we have 6 push cores from the first 3 dives. These specific rise, young basalt, not a lot of time for sediments, so we don't have any push cores from that, but we do have these from here. And Alvin goes about, let's see, 2 knots. So like, let's see, a knot is like 1.15 miles. So not very fast, like a mile an hour, basically, what Alvin does. And if you're down there 6.5 hours, you can go 3 or so kilometers if you're kind of going fast. And so all these points, they're doing science, collecting samples, looking up rocks. This part of the cruise was a rock-based cruise. They were picking up rocks on these seamounts. Okay, diffuse flow. So I'm interested in the fluids as well as the rocks. And so we saw this. This was from the video. This is where we have maybe 50 degrees Celsius water, plus or minus, coming out of the hydrothermal vent. And then the black smoker is the 350 Celsius water coming out and hitting the 2 degrees Celsius seawater as well. Okay. So these are some of the points that we have. This is temperature on the x-axis from 0 to 400. pH on the y-axis from acidic up here to near neutral up there. These are not adjusted for those of you that are into this sort of thing. And the samples that we got were from diffuse flow that was less than 100 degrees. And then we got some samples from the smokers that were up here, 350, 375 Celsius as well. So we have splits of all of these samples here at Montana Tech, and scientists all around the country have other splits of these samples as well. The upper temperature limit for life, 121 Celsius, right about here. And so I was hoping that we would get some samples that were closer to that limit, but you have to go with what you can find down there. So these are some of the samples that we have. Okay. Oh yeah, I wanted to show you this Elven window because it's really awesome. So that is the window of Elven. And so there are 5 windows on Elven. There are 3 out the front and then 1 on either side. And so I was the starboard observer, which is the right side of Elven, if Elven is facing you guys. And so I would look out this window here, and then the window kind of up in front of me as well. The pilot has the middle window, and then he'd be like, Alysia, look for black smokers out there. I'm like, all right, I don't see any, we're okay, out my side. But that's the window. And so the big part is outside, and remember the pressure is pressing those in as well. So they kind of go in, and this is after our dives, they were taking Elven apart and cleaning everything up and making sure everything was okay. The rule was don't touch the window. That was the rule. So you had to remove all jewelry and things because they don't want anything scratching the window. You see it's really, really thick, but that I think is what they consider the weakest point in the sphere are these windows. So they're acrylic, very, very thick. And so you'll see when I'm taking pictures, I was kind of far away from the window, because I didn't want to touch it and get in trouble. And then the pilot, he's used to it, so he took a picture, with the camera right up to the window because he's a pro. Okay. All right. So now I'm going to tell you about our particular dive, Alvin Dive 5007. So 5007 is actually what it means. So Alvin has bed down 5,007 times. Remember, the first subset was Alvin 1, and then Alvin was refitted for the second bit. Every time Alvin goes down, 3 people. 1 pilot, 2 science observers. Every 7th dive is a pit dive, pilot in training. And so on that dive, there is a pilot, the pilot in training, and 1 science observer. So we had 1 pit dive on our 8 dives, and so the Dan Furnari, the professor, scientist emeritus, went down with the training pilot to collect rocks. He collected a lot of rocks that day. So, all right. So to do this dive, we worked together as a team of 11 scientists. We split out our 4 different dives, and the objectives for our dive here were to do a bunch of work up there where it says flee, actually near M-vent. We were going to collect a bunch of diffuse flow, and then we had to deploy some crab traps around Teddy Bear, and we were supposed to scope Teddy Bear and go to Tika Crab Spa. We had to deploy a high temperature thermometer in P-vent in Bio 9, and then over here we were taking images of basalts slightly off axis from our downward facing camera. So this was our plan. This map was made by Sentry, so it's very detailed, and then our path was superimposed on it after the fact. Okay? And we followed our plan pretty well. So I had picked a point for us to go down, and we descended there, and that was very close to our plan. You notice a little bit of looping around Teddy Bear. We weren't quite sure where we were, and then we're like, oh yeah, that was it. Turn back around. That's what that loop is. Okay. All right. So we're going to look at our dive in 1080p footage here. All right. Let's start us back here. All right. So we've just descended for an hour into the depths, and we made it to the bottom. And then we are starting to go along to our target, which is M-Vent. And we see all of the basalts. We're kind of looking around. We're getting oriented, and we're zooming towards M-Vent. These pictures, I think, were 1 every 5 seconds. So we're going to see our 6.5 hours in like 8 minutes, maybe. That's M-Vent. It's labeled. So I think Stefan Sievert works in the same building as my co-science observer, and she's like, oh yeah, he says it's labeled, so it's going to be fine. So this is an area where there's actually been long-term research. It's called Ridge and Inter-Ridge. So all sorts of scientists have worked together, geologists, chemists, and microbiologists. Okay, so this is a temperature probe, and this is a tube for collecting water samples when the filter is on. So we are looking for diffuse flow to collect our samples with a nice temperature that everybody wants. So we're going around, and as well as our pilot was Danik Forsman, and there's a crab. Oh, this is cool. See, ooh, the crab attacked the thing, and the pilot had to like shake the crab off. Okay, look, here we are. This is M-Vent, M' vent, and there's our sample. So we actually do a whole bunch of sampling here. I called this crab condo because of these crabs were crawling in and out, and so everybody started calling it crab condo, so that's cool. Yeah, I like that. This is a major sampler. So this, I think, costs... that one of them failed. So it's going to deploy. The springs are going to come out if they work. One of the sides failed on that one. One of the other things we were doing is we were trying to find a spot that the pilot could have the sub in the same spot for a long time. So some of those other places that we were testing, there was too much current to stay in the same spot. So this spot was good. We had diffuse flow. We're collecting samples. This is a gas-tight sampler as well. And then, oh, there's the rock. Oh, we collected the rock. You see that?
Did you whack it?
Yes, that was an accident, actually. but we were excited because we're like, oh, that rock is correlated with all of our samples. Perfect. And it fell right into the basket, too. So it was like, all right, our sample, there it is. And so now we're doing a bunch of filtering of this for filling. It's actually a wine bag. And then the water's filtered for microbiology here. So there's a whole bunch of filtering happening. We're hanging out here filtering. And you can see the crabs moving around. Okay. So we did a bunch of sampling, and then there we are. Okay, so we're going to zoom around and take some images of the area for the record.
How long were you there?
At M-Vent, a couple hours maybe. I think like an hour and a half to do all that. So when we're down there, we're taking notes on papers, on sample sheets, and we have video or voice recorders. So we were clicking in and saying, report the time, location, and then observations. And all of this gets written up in a cruise report that gets put together by all the scientists. All right, so now we're zooming south here and looking at all of the cool rocks. We're going to see an octopus in a minute. And we kind of went, whoa, an octopus, and went over and looked at it. Took a little video of the octopus. But here we are. So we're in the trough, and we're traveling along the trough for the good views as well.
How far off the rocks are you?
Oh, so there's the octopus. So for this down cam that Dan Furnari has on the bottom, he wanted us to be, I think, 10 meters up a little bit for traveling. And so we were basically traveling however Danik thought, our pilot thought was a good distance for traveling. This is a diffuse flow vent. We have crabs, we have little shrimp, we have tubeworms as well. This is the slurp. This is basically a vacuum tube. So now our mission is to collect those crabs. So if you'll notice, we're sucking up crabs, and they're going into the slurp. And those crabs are for one of the biologists on board who was doing some work with these crabs. Okay. So this was the next part of our mission, was collect crabs. And then the slurp jammed. Like it got a rock in it, and so we stopped using the slurp. So I think we collected, the numbers. I think we got 7 crabs that way. I'm not sure. And then it's going to end up jamming a little bit. And then we're moving on around. And we can see the really cool basalt formations as well. More diffuse flow. All right, so now we're deploying the crab trap. And we actually found out that, oh, look, the crabs go right for the dead riftia right away. And so we're doing some other stuff. We were deploying microbe traps and taking some temperatures as well. There's another cruise to this area sometime next year. They're going to pick up these microbe traps for the scientist who's in Charleston. And she's going to study what grew in them for the next couple months. So now we're doing biology stuff. And the cool thing that Danik figured out was, oh, if we want to catch crabs and our slurp isn't working, we just pick up the crab trap and we shake it into the box, because the crabs were going at the stuff, at the riftia, like on the outside of the box. So he actually was like, oh, cool, look at that. And we got some crabs. So we actually did a good job getting crabs for Carolyn Teepolt. She's an assistant biologist at the Woods Hole Oceanographic Institution. She does some interesting work. All right, so that was our mission there, was to get some crabs and to deploy that crab trap and to deploy the microbe trap. Now we're continuing on, moving south. There are some other, one of the scientists was doing heat blankets. She's trying to figure out the flux of heat. And so I think we see one that was deployed, and we kind of drive we drive over it to get an image of where it was. All right, so now we're getting close to Bio 9 vent, and we have a look at it. There's a little bit of smoke as well. So this camera is from the center of Alvin, from the brow. There's some black smoke as well, and then we're supposed to deploy this thermometer. We actually deployed this wrong. That is not the way to deploy it. We got in trouble for this. So it needs to be hanging in the vent, and this needs to be out, because if it's sitting there, it can get grown up around it. But Danik couldn't get it to stick the right way, so we left it like that, and then the next day they were planning to go to that area anyway, and then they fixed it and got it in the right spot. So there we go. Black smoker. Now we're collecting a sample in the plume. So we're having the front of Alvin in the plume. One of the scientists was interested in the plume. We have some plume samples as well. And so the Niskins are getting deployed. So we're getting a discrete water sample of the plume from the hydrothermal vent as well. And then there we are, off, driving over the basalt, looking at the really cool rocks. At this point, I think one of our hydraulic arms, we stopped using it because it looked like there might have been a leak from it. And so if you see that, you just stop using it. We were supposed to collect a rock, and the geologists that wanted the rocks were annoyed with us because we didn't collect the rock but it was the arm, basically. So they got the rock the next day. So it was okay. So there we are.
Do you have to pee yet?
Excellent questions. So there are pee bottles there. There's like one for everyone and then some extra. I stopped drinking fluids the night before at 11 so that I wouldn't have to pee. And then by the time I got up, I really had to pee. And on every single tour that I gave, somebody always asked that question, for sure. Yeah. But apparently everybody's very professional, because basically you have to pee like right behind, like everybody's like right there. So yeah, so we all tried not to pee. Okay, that was the Brow GoPro camera video. All right, so now I want to show you the pictures that I was taking out of my window and how it compared to other pictures. So here I am, not touching the window with the camera and being like, whoa, this is so cool. Here's the slurp. This is the starboard arm. The starboard arm, I guess, isn't as good as the port arm. So the port arm does a lot of the work, and the starboard arm has a camera on it and does less work. And I think that's just how it's been engineered. Okay. The pilot, so Danik takes this picture out of the center window. He's bold and can, get really close to the window and not be afraid of touching it. And so this is what it looks like from the center window. And here's our rock, again, that we have up here. You can see a little bit of the major samplers haven't been deployed because the springs are in here. The Tom Thumb is one of the hydraulic arms on Alvin, and it comes in and it presses this down, and then that activates these. And then they the springs expand and suck the water in. And then there's a little bit of a microbe trap. So that's the pilot's view from the center window. And then this is a little bit fuzzier. So this is the port brow cam. So there are cameras all over Alvin. And as an observer, you have a little screen in there, and you can toggle through all the cameras. So you can look at whatever you want to, and it also records one set. So we always had one recording from the middle camera, and then we switched the other view for whatever was going on at the time, whatever had the best view. And then those are all recorded and go in the archives, and you can click through and see all of those images that we took as well. So I could click to any camera. I actually spent a lot of time looking out the windows and doing some of the camera stuff because it was interesting to look out the window. So you'll notice this is a little bit fuzzier because it's not a GoPro or 4K camera here. All right, that is the difference. So Dan Furnari of the MISO, which is the Multidisciplinary Instrumentation in Support of Oceanography group, has been getting all of these cameras rated and integrated into the Alvin system so that they can actually take cameras or take images and video that looks like that. So just like the really nice imagery that you saw, the video that we saw, that's what Dan is going for with these MISO GoPro cams. Okay, so that's our rock again, right down there at Crab Condo near M-Vent. There's the octopus from my view, out my window, so there he was. So if you can imagine, living your life on the seafloor, it's super dark, it's really chill, and then all of a sudden this huge white thing comes out of the mist and shines lights on you. Well, that's what he was experiencing, or she was experiencing at this point. Did not bat an eye, did not move a tentacle, just sat there while we shined lights on it and took pictures. It's pretty chill octopus. Okay. All right, and this is another image of the diffuse area. So we see a lot of those shrimpy things. We see the riftia, the tubeworms. Some are out, some are in. We see crabs. Oh, that's a squat lobster. That's a squat lobster right there. So see how its shape is a little bit different than the crabs. Oh yeah, the technique for slurping the squat lobsters is from behind, and then you slurp crabs from the side because they're less distance, yeah. So you see the red on top? That's just the reflection of the interior sublights on the window, so as well. All right, this is one of the pretty good images, I think, that kind of summarize a lot of the things that we did on Dive 5007. So besides collecting all of those fluid samples and this lovely rock here, we deployed crab traps, we deployed microbial traps, we collected a bunch of water using the tubes, and that is a good image of that. And then there's the rock again. And then here are the major samplers. So yellow, black, blue sampler. And then here's that temperature logger that we deployed as well down there. And each one of these is a discrete sample, and we filtered this all into different things. So we have a bunch of these samples all preserved and split out amongst the geochemists that were on board and on shore as well. So that's also representative of the samples that we took. You can see from this one the basket. So here's one of the baskets for setting things in as well. Okay. All right. So we're going to look at, this is the camera below Alvin that is taking a picture every 10 seconds from below. If we watched it at full speed, you'd probably throw up. So I have to pause it and slow it down a little. So what I press this. Yeah, okay, there we go. Now nobody's going to throw up. Okay. No, not one. Just kidding. Half speed's good. Okay. A little slower. Yeah. All right. So this is taking a picture once every 10 seconds while we're doing work and then while we're moving. And I actually went back through all that stuff was us setting up kind of some iron oxides. You see diffuse flow where there's some sulfur. There's a whole bunch of iron oxides down there as well. We flashed by some really interesting anemones that look like snowboarder hats. Us, uneducated geochemists called them snowboard hat anemones, and I forgot the name now, so I can't tell you the real name of the anemones. But there are some really great pictures of rocks in these downward-facing images. They're really good. And then every so often you catch the occasional creature. That was an anemone that flashed by. It was really, really fast. We turned this off sometimes while if we were sitting on the floor. And I think the best focal length for this is, I think, 3 or 4 meters up. Yeah, so some really good images of rocks. So these are all kind of strung together. There's some more diffuse flow going over that area. Here we're sitting down and we didn't turn it off, so it's taking pictures of right below us. There's an anemone, a red one. Lots of crazy stuff on the seafloor. So it was really interesting, to go through the dive with my window, taking my notes and like doing my recordings, and then going back through and looking at what we were seeing below us, and looking at the view from the top, from the middle front as well. So lots of really great pictures of basalts as well in the area here.
How does Alvin know where it is?
That's an excellent question. Yeah, so actually, they have all of these coordinates from all of the dives before. And the reason why we did that little loop at Teddy Bear was because we were at the exact coordinates from the dive before, but it wasn't exactly kind of lined up. So Alvin actually sends signals like up to shore or up to the ship about what the exact location is. But it is unclear to me why it was not exact compared to the cruise before. So they have all of the coordinates. There are 2 different coordinate systems that are used here. So one, because there was a lot of work going on in the area, the researchers set up a grid and they used that. And then some of them are just using lat long as well. Oh yeah, so did you see the one go in and out? Yeah, cool. Yeah, look at that. It's pretty cool. So that one's going in and out. Yeah. So while we were down on our dive, we actually talked to the American Geophysical Union meeting as part of an outreach event. So Adam Sewell, one of the heads of the Deep Submergence Facility, who takes care of these vehicles, beams down and talked to us. And I don't know if it was the most intelligent thing I've ever said. I was like, "This is the coolest thing ever," And then the guys the next day were kind of prepared, and they said something, like, "Oh yes, we're down here for," But it was pretty good. I guess everybody appreciated our enthusiasm, for sure. So more basalt. Yeah, but it is, it's all very quiet, and those submarine movies that you watch with like the pinging, it's kind of a little bit like that. All right, so this is going over a place called Riftia Mound. So we flew over where there's a huge, huge mound of Riftia, and we had been doing a bunch of work over there with those crab traps and collecting things, and now we're going over to the Bio9 and P vents and doing the collections in the plumes and deploying that thermometer. So that's what's going up above while these images are happening below as well. So yeah, hour down, about 6.5 hours on the seafloor, and then we drop weights to come up. So we're carrying plates of steel weights. So every single Alvin dive, plates of steel get dropped on the seafloor. And then we go up like that. And I guess that's the cheapest way to do it, is to use steel weights as well. So here we are. We did our work there. Now we're going off axis a little bit, getting some cool pictures of some basalts. And then we sit a little bit and then we head back as well. When you see that fluff, we've agitated a set of, we've kind of set down in some sediment. All right. Well, I think that is getting very close on time. I have a couple of images that I will show you relating the East Pacific rise. So this is some of the work that we do in the lab. We sample, so temperature and pH again, a wide variety of samples. The pink ones are from the Andes, from a subduction zone. And the East Pacific rise are the purple squares as well. And this is human blood at the top X, your stomach at the bottom X. And we looked at samples from here, but remember that those are diffuse flow samples from that part of the graph. But remember that these black smokers are way up over here. So we smash everything down into less than 100, and because of the pressure, we can have these very high temperatures. Okay, black smokers. So we're linking microbial activity with geochemistry in general in lots of environments, and you heard about Alvin Dive on the upper left. The chief scientists were Trish Gregg and Dr. Dan Furnari, and then PIs Mike was on board, Dorsey was not on board, Barbara John was on board, and Ken Costle was doing communications. So lots of science team people, and thanks to all of the funders for this, and the crew, and the science Alvin and Sentry operations groups as well. And with that, I will take questions that you have. Okay. Yes,
So
please.
When they're using the slurp to get the crabs,
Yeah.
are they keeping them in something that keeps them in a pressure? When
Okay, so no. So they're actually slurped into these little vesicles that rotate. So it's just like a little container like this, and they hang out in there. And you can't slurp certain organisms with other organisms, or they like eat each other. So you can't slurp an exoskeleton thing with a soft thing, where the soft thing disappears. But yeah, but on this cruise, they did not have pressure-rated bio boxes. On some, they do. So people that are, because the early career scientists, they don't have the fancy pressure bio boxes. So
what
no.
happens when they,
So the crabs go from 250 bar pressure to sea level, and they're stressed out, but they're okay. The tubeworms, they're dead. They don't make it up. I was too. I was pretty shocked that the crabs were okay. So crabs are pretty tough. Yeah.
exoskeleton works?
Exactly. Go for the exoskeleton. Other questions? Yes. How is Elven powered? Okay, so all of that, a bunch of batteries and all of that, yeah, is powered. So yeah, not tethered. It's free. Yeah, and yeah, the batteries are rated for something like 3 times the amount of time that you need it, and there's air down there for like 3 days in case something goes wrong, and nothing has ever gone wrong with Alvin. They've had like an impeccable track record since 1964, so I wasn't worried. Yes. Yes, the back.
Do you find any interesting microbes growing in the high pH and the pressure and the depth?
Yes, there are very interesting microbes that are growing down there, and people, so we have some samples here to look at that, and then some of the other microbiologists also have samples to look at those. There are, yes. And they colonize first, so if there's an eruption, then the microbes are the first thing to go there, and then they start to set the stage for the larger organisms as the system recolonizes after each eruption, approximately every 10 to 20 years.
Did you have to undergo training prior to getting in this?
Yes, we did. So we underwent, so before this, I think I was accepted to this. I applied like the day before it was due. Well, actually, no, I turned in my application the day it was due because I was like, oh, I should probably do this. And I wrote it and sent it in. But so that was like May or June. And then we started having meetings and we did webinars and we planned things all the way up until December. And then on the ship, we were trained. We got the safety, and then we got trained on putting on the carbon scrubbers in case the air went out as well. So we are less trained than the pilot. The pilot, he told the process. So our pilot was the youngest Elven pilot ever. He was either 29 or 30, and they went through. They have to be able to talk to scientists. They go through a panel of scientists to get approved, and then they go through a panel of like engineers, and then they go through the Navy panel. And so they ask you all sorts of serious questions, at the Navy panel to make sure you can, handle yourself as a pilot. So pilot, highly trained. Science observers, less training. Yes. Yes, please.
What was the temperature of the water when Alvin was going through the smoker?
Oh yeah, so we couldn't go through, Alvin's guts and body were not in the smoker plume, but the front basket, we could put the front basket in the plume to trigger the Niskin bottles. So it was probably more like 60 Celsius-ish when it was in there. So yes, the Alvin cannot go in any of that hot water. So it's probably one of the most dangerous places actually for Alvin to go is around the high thermal vents. Our pilot was like, oh, this is the coolest place ever though. So, and it was pretty much the coolest place ever. Yes.
Would there be as much animal life if you went out, say, 10 kilometers from
No,
smokers?
not quite as much. So at the beginning of this, if you came in early, I was kind of playing some of the images from the Seamounts. So there was, some animal life, but these ecosystems are hosted right there at the black smoker part, and there's less as you go out. Yes, good question. Nick?
Is that species of crab that's found down by the black smokers?
Yeah.
Or is that found, like, I
So-
go to San Diego and see it?
So I actually talked with the crab biologist a whole bunch of, but I think it starts with a G. I think it's Gallinella, something like that. Starts with a G for that crab species. And so she was doing experiments on these crabs and compared them to coastal crabs, and they actually have a different stress response rate to changing conditions than coastal crabs do. So I don't know the answer to your question. Not sure. Probably not.
Did
but-
Alvin view eruptions?
So yes, so not during an eruption, but Alvin in April of 2006, I think that's right. So after that other eruption, they went down with Alvin in this area and they were like, oh my gosh, there's been an eruption. So Alvin has not been down during an eruption, but very soon after the eruption. Because I think they decided that it started erupting late 2005, and then they went in April 2006 and it had been subsiding. Yeah. Yes.
The solids that are coming out of the vents, do those just fall to the seafloor, or are they carried?
Yeah. So there are a lot of cool papers on the plumes and the distribution of plumes and how far you can trace the plumes out, actually. Yes. So send me an email and I can hook you up with some papers on that topic.
Thank you.
Yes. Other questions? Yes.
Is the cool seawater that surrounds these things?
2 degrees Celsius, yes.
Is that oxidizing or reducing?
Oxidizing. Yeah. Yep.
Surprised.
Yeah, there's still oxygen, like all the way down. Yeah, through the water column. That was like my general exam in chemical oceanography, like right there. Draw an oxygen profile. Yeah, okay.
Where are you going back?
So we're gonna write proposals that are due August 15th. And us, of us 11, 4 of us are gonna each write one, and then when 2 of them get denied, then the other 2 will go on with the other one. Yeah, exactly. So, and then ships get scheduled and et cetera, et cetera. So then like a year, so it takes them 6 months to review that, and then like a year or so after that. So we'll see. We'll see.
What's the late career scientist?
Well, you can get. You guys can get on my assistant list because I've already got a long one. Yeah, Chris.
I was just curious about the 2 degrees because I don't know, is that the maximum density for seawater?
Yes, it is. Yeah. Well, it's right about there.
Pretty close.
Yeah, but it's very close because it's 4 degrees C for fresh. So, yeah. Yeah, there's a lot. I didn't talk about any of the cool chemistry that happens down there, but there's a lot of sweet, cool chemistry that happens at those temperatures and pressures with seawater and hydrothermal vent fluids. Yeah. Yes?
Did you ever figure out what minerals were associated with the microbe growth first
Like
after an eruption?
You said that
the
so
ecosystem, like what? Were they-
hydrogen sulfide, those are basically off of hydrogen sulfide. They fuel most of the system down there. Yes. Other questions? Okay, I've exhausted you. So come up and have a look at the rock, and if you haven't seen the tubeworm, come see the tubeworm. If you have any questions or want more information, I have my card here because I don't have all this up on my website. I'm sorry, but I can get you information. Okay.
Thank you.